Assalamu alaikum. This is an introduction to immunology by me, Dr. Sara Abu Zaid, PhD in immunology from Damietta Faculty of Science, Egypt. Here the second lecture, Innate Immunity, Part 1. Taliaferro said the host is an island invaded by strangers with different needs, different food requirements, different localities in which to raise their progeny. There are a formidable range of infectious agents that can use the human body as a sanctuary to raise their offspring. The immune system faces the task of providing a defense mechanism to establish a state that is known as immunity to infection. Definition of immunity is therefore all those physiological mechanisms that endow the animal with the capacity to recognize materials as foreign to itself and to neutralize, eliminate, or metabolize them with or without injury to its own tissues. Immunological responses serve three broad functions: defense against microorganisms, homeostasis, removal of damaged or effete cells, surveillance, recognition, and destruction of mutant cells. These responses have been widely classified as non-specific and specific functions. Among the non-specific defense mechanisms, there are those that form a set of ill-understood and perhaps grossly underemphasized constitutional factors that make one species innately susceptible and another resistant to certain infections. Constitutional factors that control non-specific defense mechanisms include genetic between species, age, metabolic, neuroendocrine factors, and environment. Genetic control of disease has been shown to be strongly associated with the major histocompatibility complex. For example, here we see Mycobacterium leprae seems to infect humans and armadillos only. We can see Bacillus anthracus is an infection of humans, though not of chickens. Norrea is a disease of men and champagnes and not of any other species. Some species seem to be able to harbor organisms within their body tissues, while the same organism may cause another species to succumb to such an infection. Within men, there are certain well-known racial differences in disease susceptibility. Dark-skinned individuals have an increased susceptibility to coccidioidomycosis type of fungi. Certain dark-skinned people lack the red cell Duffy coat and are not susceptible to vivax malaria. So we can see the genetic control of disease and its relation between species. Age. The spectrum infections and certain viral infections of children are more severe with age. The very young are more susceptible to many infections, in particular Escherichia coli meningitis. This may be because bactericidal immunoglobulin M does not cross the placenta. Metabolic factors. We can see hypoadrenal and hypothyroid states decrease resistance to infection in diseases such as diabetes mellitus, where altered metabolism causes increase in blood glucose, decrease in pH, and reduced influx of phagocytosis and of phagocytes. Infection can be a severe complication. Also, steroid hormones are known to affect many modalities of the immune response. Neuroendocrine factors. There is a growing body of evidence that supports the hypothesis that immune processes can be influenced by neuroendocrine factors. Immunologic cells have receptors for a whole range of hormones, corticosteroids, androgens, estrogens, and progesterone depress immune responses, whereas the growth hormone, insulin, and thyroxine do the opposite. Immunologic organs are innervated by autonomic and primary sensorial neurons. Hormone secretion is balanced by neural control. If therefore seems reasonable to say that immune responses are finally tuned by neuroendocrine circuits at a more physiologic levels, stress and circulation rhythms modify the functioning of the immune system. Environment, poor living conditions, overcrowding, and undernutrition also increase susceptibility to infection. This is constitutional factors that make one species innately susceptible and another resistant to certain infections. Natural barriers to infectious agents. Natural barriers to infectious agents are simple yet effective means of innate immunity. We can see here in the respiratory system, the cilia inside the respiratory tract. We can see mucus and the cough reflex. In the digestive system, there is mucus, there is acid in the gastric juice, there is intestinal enzymes and colonic flora. In the skin, the intact skin itself, it is a natural barrier, okay? The sweets, the sebum. First, intact skin. A major form of defense in this context is the intact skin, which is impermeable to most infectious agents. Sweet and sebaceous glands are potential points of entry for the infectious agents. However, most bacteria fail to enter due to the low pH and direct inhibitory effects of lysozymes, lactic acid, and other fatty acids of sweet and sebaceous secretions. An exception is Staphylococcus aureus, which commonly infects the hair follicle and the glands. Here we can see this video illustrating the immune nature of the skin, and we will later explain these cells.
The skin is the body's primary barrier against physical insults and microbial pathogens. It represents a unique environment in which immune cells interact with skin cells to maintain tissue homeostasis and induce immune responses. The skin is composed of epidermis, dermis, and a subcutaneous fatty region. Commensal bacteria, fungi, and viruses living on the skin have beneficial effects in the protection against pathogens and in wound healing. The epidermis is composed of highly specialized epithelial cells known as keratinocytes. They are continuously replenished from just one layer of basal keratinocytes, which divide frequently. Dead cells called corneocytes form the outermost layer and are largely responsible for the barrier function of the skin. In the dermis, cells known as fibroblasts secrete elastin and collagen fibers that form a dense extracellular matrix. Blood capillaries irrigate the dermis, while lymph fluid is drained through lymphatic vessels to lymph nodes, specialized immune structures in which immune cells are activated after pathogen encounter. Diverse and functionally specialized immune cells populate the skin. In the epidermis, a specialized subset of dendritic cells, called Langerhans cells, sample antigen. They project dendrites upward towards the cornified epithelial layer and sample bacterial antigen such as toxins. Langerhans cells appear to be both anti-inflammatory and activatory depending on context. Dendritic cells in the dermis are highly efficient at capturing dead cells and presenting antigen, such as viruses, other intracellular pathogens, or skin-associated self-antigen, to T cells. If dendritic cells are the immune sentinels, T cells are the immune effectors. Healthy skin contains more than twice the number of T cells found in the blood. Most of them are memory T cells that have previously encountered antigen and can be rapidly reactivated. T cells in the epidermis are mostly CD8 T cells, a subset that becomes cytotoxic and kill target cells upon activation. They're long-term residents in the epidermis, mostly disconnected from the circulation. T cells in the dermis are mostly helper CD4 T cells, which have a more modulatory role in the immune response. A variety of other immune cells, such as natural killer cells, eosinophils, and mast cells, are present in the dermis and might be involved in allergic reactions in the skin. Dendritic cells and keratinocytes sense tissue damage, such as wounds or cold sore lesions that occur when latent herpes virus reactivates, and they do that through evolutionarily conserved receptors that recognize pathogen-derived molecular patterns or host-derived molecules that are exposed by cell death, such as DNA. Keratinocytes produce antimicrobial peptides, which can kill bacteria directly, inflammatory mediators, such as interleukin-1 or IL-1, which activate dendritic cells, and chemokines, which recruit neutrophils, macrophages, and T cells. Activated dendritic cells migrate to the lymph nodes, where they present antigen from the site of infection to naive T cells, priming them to activate and differentiate into effector T cells. Activated T cells return to the skin and kill infected keratinocytes to control viral infection, or secrete signals that recruit additional immune effector cells. Following viral clearance, memory CD8 T cells persist in the epidermis to provide immunity for future encounters with the same virus. Immune responses can become dysregulated and cause skin disorders such as psoriasis or atopic dermatitis. Psoriasis is a lifelong inflammatory skin disease characterized by scaly reddish plaques. A combination of environmental and genetic factors confers susceptibility to the disease. Physical injury or inflammation can trigger the formation of an acute lesion. The antigenic trigger is unknown, but current models propose that stressed keratinocytes might release self-DNA, which in complex with an antimicrobial peptide activates dermal plasmacytoid dendritic cells to secrete high amounts of the antiviral mediator interferon. Together with pro-inflammatory IL-1 alpha released by stressed keratinocytes, interferon activates dermal dendritic cells to promote T cell differentiation. The earliest recognizable change in the affected skin is the accumulation of T cells and dendritic cells around blood vessels in the dermis. An overt lesion occurs when CD8 T cells, dendritic cells, and neutrophils infiltrate the epidermis. Specialized subsets of T cells secrete soluble mediators like interferon gamma and IL-17, which stimulate the proliferation of keratinocytes, and this produces a marked thickening of the epidermis. Signals from the proliferating keratinocytes act as chemoattractants for infiltrating neutrophils. Crosstalk between immune cells, keratinocytes, and dermal cells thus contributes to tissue remodeling and amplification of this dysregulated immune response. Without treatment, acute psoriatic lesions become chronic lesions. Genetic studies have identified psoriasis-associated susceptibility genes, some of them linking Th17 cells, the subset of T cells that produce IL-17, to psoriasis pathogenesis. Thus, immune cells in the skin exert important roles in maintaining the barrier function against pathogens, but can also become activated by self-antigen or harmless antigen to cause autoimmunity or allergies.
Mucus secretions of the tracts that connect internal organs to external surfaces form an important form of defense. They entrap and immobilize bacteria and hence prevent adherence and colonization of epithelial surfaces. Mucus secretions we can see in nose, in the mouth, in our external surfaces. Let's see this explanation.
If you've got a cold, mucus is hard to miss. But what is it? And what does it do besides making you miserable? Your body produces more than a liter of mucus every day. And all the wet surfaces of your body that are not covered by skin, like your eyes, nose, mouth, lungs, and stomach, get a liberal coating. That's why they're known as mucous membranes. Mucus plays lots of roles in your body. It keeps delicate tissues from drying out and cracking, which would expose them to infection. It lubricates your eyes so you can blink. It protects your stomach lining from acid. It neutralizes threats by removing or trapping substances that could make you sick. And finally, it houses and keeps your body's trillions of bacterial inhabitants, your microbiota, under control. Mucus contains lots of different compounds, including proteins, fats, and salts. But a key component of mucus versatility is a set of proteins called mucins. Mucins are the primary large molecules in mucus and are essential for giving mucus its slippery feel. They belong to a class of proteins called glycoproteins, which are built out of both amino acids and sugars. In mucin, long chains of sugars are attached to specific amino acids in the protein backbone. The hydrophilic sugar chains help mucin dissolve in your body's watery fluids. Mucus, which is up to 90% water, stays hydrated thanks to these sugar chains. Some of these mucins can interact with other mucin molecules to create a complex network that establishes a barrier against pathogens and other invaders. That's why mucus is the body's first line of defense against foreign objects like bacteria and dust. It's continuously produced to clear them from the respiratory tract, like a slimy conveyor belt. This keeps bacteria from getting a solid purchase on delicate lung tissue, or making it to the bloodstream, where they could cause a major infection. Many of those harmful bacteria also cause diseases when they cluster into slimy growths called biofilms. But mucus contains mucins, antimicrobial peptides, antibodies, and even bacteria-hungry viruses called bacteriophages that all work together to prevent biofilms from forming. If microbes do become harmful and you get sick, the body ramps up mucus production to try to quickly flush out the offenders, and the immune system floods your mucus with extra white blood cells. In fact, the greenish mucus often associated with infections gets its color from an enzyme produced by those white blood cells. This multi-pronged approach to bacterial management is one of the main reasons why we're not sick all the time. Even though mucus protects against the infectious bacteria, the vast majority of your body's bacterial tenants are not harmful, and many are actually beneficial. That's particularly true when they live in mucus, where they can perform important functions like synthesizing vitamins, suppressing harmful inflammation, and controlling the growth of more harmful species. So even though you probably associate mucus with being ill, it's really helping you stay healthy. Sure, it might seem gross, but can you think of any other substance that can lubricate, keep your body clean, fight infection, and domesticate a teeming bacterial population? Nope. Just mucus.
Respiratory tract. We can see in the respiratory tract hairs at the external ears, the cough reflex, and the ciliated mucous membrane of the respiratory tract. All these help drive entrapped organisms upwards and outwards, as we see in the previous video, and as this video also explain more. Let's see it.
lungs are where the body draws in vital oxygen and exchanges it for carbon dioxide. But every breath we take is accompanied by potentially harmful microbes, as well as particulates which can damage delicate lung tissue. To counter these threats, the lungs are enriched in immune cells that repulse invaders and repair injury. But this potent arsenal comes at a heavy cost. Overreactions or inappropriate responses result in diseases such as asthma. The lungs are spongy organs composed of increasingly finer tubules. First comes the trachea and the bronchi. These are then divided into the bronchioles, which are wrapped in a muscular sheath. At the ends of the bronchioles are microscopic air-filled sacs called alveoli. In the case of asthma, most of the harmful immunological events occur within the bronchi and bronchioles. The bronchial wall is made up of an epithelium covered in tiny hair-like structures called cilia, an interstitium, and muscle cells. Goblet cells scattered along the epithelium discharge a thin layer of mucus into the bronchiole lumen. The mucus is continually swept back up through the lungs by the action of the cilia. This system is known as the mucus escalator and is important for removing inhaled particles trapped within the mucus. Embedded throughout the bronchiole wall are immune cells such as dendritic cells, which sample antigens from the lumen, mast cells, which are packed with granules, tiny packages rich in histamine, and innate lymphoid cells, or ILCs. Individuals with asthma need to have been previously sensitized to a specific environmental antigen. The most common is found in the feces of house dust mites. But symptoms can also be worsened by respiratory virus infections, smoke, or pollutants. When inhaled antigens contact the epithelium, they trigger the release of two chemicals, IL-25 and TSLP, which stimulate nearby dendritic cells and innate lymphoid cells, those ILCs. The dendritic cells leave the epithelium for the lymph nodes, where they activate T cells and initiate an adaptive immune response. Antigen can also pass through the epithelium, especially in conjunction with damaging particles such as those found within tobacco smoke or diesel fumes. Some of this antigen can reach the mast cells housed in smooth muscle tissue. The mast cells of asthmatic individuals are unusual. They have more of an immune molecule called IgE stuck to their surface. The attached IgE then binds the relevant antigen, in this case from house dust mites, in a highly specific manner. When an antigen gets bound to two IgE molecules, it signals the mast cell to degranulate and spill out the contents of the granules. The granules' innards then exert their effects on surrounding cells and tissues. In asthma, the granule contents cause smooth muscle contraction and mucosal edema, leading to narrowing of the bronchioles. Many anti-allergy drugs target this key process of degranulation. Back to those T cells in the lymph nodes. Once activated, T cells enter the interstitium and team up with ILCs to escalate the asthmatic response. They secrete chemical signals that recruit other immune cells, most critical of these being eosinophils. These cells play central roles in almost all allergic diseases. Eosinophils squeeze through the epithelium into the lumen and begin releasing their own inflammatory signals, including IL-5, IL-13, and platelet activating factor. Together, these act on goblet cells, causing them to ramp up production of mucus in a process known as goblet hyperplasia. By now, the victim is in the midst of an asthma attack. As the attack proceeds, more and more eosinophils are recruited into the lumen and the mucus layer progressively thickens. The collective effects of smooth muscle contraction and mucus overproduction restrict airflow and result in the classic symptoms of asthma, including difficulty breathing, coughing, and wheezing. Multiple attacks can, over time, lead to fibrosis and permanent injury of the lung.
Other mechanical factors which help protect these tracts are washing action of tears, saliva, and urine. Many secretions contain bactericidal components such as acid in gastric juice, lysozyme in tears, nasal secretions and saliva, proteolytic enzymes in the intestinal secretions, spermine and zinc in semen, and lactoperoxidase in breast milk. Normal flora of the body can suppress the growth of many potential pathogenic bacteria and fungi by competition for essential nutrients or by production of inhibitory substances such as collagens or acid. Pathogenic invasion of the vaginal flora is inhibited by lactic acid produced by the commensal vagina flora. When normal human flora is destroyed by broad spectrum antibiotics, pathogens such as candida, it is a fungal disease, and Clostridium difficile cause obstetric infections. In the next lecture, if the organisms do penetrate the body, two main defensive operations come into play: phagocytosis and the bactericidal effect of soluble chemical factors, molecules such as complement proteins, acute phase proteins, and cytokines. Other cells may also be involved, cells that release inflammatory mediators, basophils, mast cells, and eosinophils, and natural killer cells. So I hope you enjoy this lecture, and see you in the next lecture. Thank you.